Sure, everybody, welcome. Welcome to Lincoln University. Welcome to this beautiful building called myriad. It is our latest addition to the Lincoln University campus. Yeah, if this is going to be the standard, then we'll be really, really, really happy. Welcome also to the Excellence Series. And how it's going to run today is that I'll do a little bit of housekeeping. I will introduce you to our provost, Professor Chad Hewitt. He will then be your master of ceremony and introduce my dear friend, Professor Rainer Hoffman. And then we'll just let it run. Afterwards, we'll have some drinks and nibbles, but I'll talk to you a little bit about that later. So this thing should work. Was it me or was it you? I think it was me. Cool. Oh yeah, it was me. Housekeeping. We had a fire test this morning. So if we hear the fire alarm now, it is the real thing. What we do is really easy. We see the exits at the back, and if there's no fire out there, we just go out the back doors and we get our own Forbes lawn. So that's the greeny bit that you guys see out there. If there is danger out there, just follow us, and we'll guide you through emergency exits to the other side of the building. Toilets are just outside this door, and refreshments. So after Rainer has talked, there is time for refreshments and a nibble and a good discussion. We will be serving some alcoholic beverages, so please make sure that if you drink those, you think also about getting home safe, because we know for sure that the people that are sitting home waiting for you want you home safe. If you want a soft drink, there is a little bit of a new procedure. You need to walk to the fridge, you need to pick out your soft drink, and you need to show the ladies from Lincoln Hospitality what you picked out of the fridge, and they will actually put it on the list so that Chad gets the bill. All right? Cool. Yeah, without any further ado, I think I'm going to introduce our provost of the university, Professor Chad Hewitt, and he will take it from here.
Jim. Good afternoon. I'm Chad Hewitt, as Jim said, provost of Tafade Wanaka or Aotearoa, Lincoln University, and I'm pleased to welcome you to the 8th in our series of excellence lectures. The Excellence Series is Lincoln University's element of contributing back to our community, and specifically to contribute deeply to our public debate of the many critical issues facing our country and our communities, while also showcasing the university's world-class research and education. Rainer is a recently promoted professor who is providing us a significant insight to his research. His information about how sustainable plant innovation can change the world. And the presentation will showcase several plant-based approaches from New Zealand and overseas that are part of climate-smart, sustainable intensification practices in agriculture. That's what you're going to talk about.
Absolutely.
Yeah. Rainer came to New Zealand from Vienna, Austria in 1993. He's a plant physiologist and professor of plant biology at Lincoln University. He's also acted as an interim dean and head of department within agriculture and life sciences. He works in a branch of science that studies how plants work. He uses physiological and biochemical tools to examine plant-environment interactions with a key focus on plant stress resistance. Most of his work is related to climate change resistance. To address this challenge, he worked closely with molecular biologists, plant breeders, agronomists, ecologists, social scientists, and related disciplines. His research includes a number of national and international collaborations, including with CRIs, industry and other universities in New Zealand as well as overseas. Regularly cited in UN reports, he has a special research interest in sustainable agriculture projects in the Pacific Islands region and has developed strong agricultural links in the Pacific Islands with tertiary and development partners. Rainer Hoffman is a New Zealand Institute of Agricultural and Horticultural Council member and a recipient of a National Agroecosystems Sustained Tertiary Teaching Excellence Award. Welcome, Rainer, and I hand it over to you for the next 35 minutes.
Thank you. Right. Kia ora. I will talk today in a whistle-stop tour pretty much about all that work that I've done since I've arrived at Lincoln Uni. So it'll be quite a few slides, and I apologize to the non-scientific members of the audience for some graphs that have error bars in them, and just don't worry, the errors are part of life. And that's really the theme of today. It'll go quick, it'll be over in a heartbeat. Okay, so the first picture I want to show you, if it counts.
Yeah.
Is it there? Is from Dusky Sound. Who has been at Dusky Sound? What would you say is particularly stunning there?
Prehistoric.
Prehistoric. That's a great word.
Jurassic.
Jurassic. You walk through this track, and so this is in the southwest of Fiordland, and in that year when I was there, it was 1988, and there was massive spider webs, and you had to. The first person in the team was the spider catcher. You were like this, walking on the track. It was at times really prehistoric. That's a really good description, and that roped me in because before I had been in that environment, blessed by it, I was a medical student, really unhappy, not really into it, and an environmental campaigner in Austria. And so I was still on the search. This is where I got captured by your environment, and this is why I became a plant scientist. And that's why I show this picture at every lecture series when I start and tell my students that it's too precious to risk. It's full of diversity of memory, millions of years of evolutionary wisdom, and so let us learn from that. So a lot of the talk is about diversity and what to make out of it. Another inspiration for me on the human front was this man, Albert Schweitzer. Who has heard of him? What's your recollection?
He was a Belgian who worked in the what was then Belgian Congo as both a missionary but a hospital and rural development type work.
Yes. So a humanitarian and also a philosopher. And he developed this insight about life that he called the reference for life. And it basically means complete and utter respect for the diversity on the planet that includes plants, animals, people, and he's really lived that as someone who got a Nobel Peace Prize for his work during his life as a humanitarian, where he built hospitals in Africa, but he also wrote a lot of philosophy about what life is all about. And that captured me to the point where I now realize I'm actually, living this vision of life, which is basically around respecting the differences that we have, and actually embracing them. so that the truth, whatever that is basically a combination of many different viewpoints. And that means you need diversity, and you need actually quite contrasting perspectives or aspects in systems to get to a point that approximates something called the truth. So that tells you a little bit about where I'm from in my journey, and I'm not now gonna use that introduction to really home in on this reference for diversity. And in my case, it became a journey in the world of plants, thanks to Fjordlands, thanks to Dusky Sound, in the agricultural world of plants, which means that you can use that appreciation of diversity in many different aspects, and I'll give you examples for that. This is from a paper that was published recently in Nature Food, and it basically says at the base of resilience, and this is what we need to think about, isn't it? Climate change and population growth. At the base of resilience is diversity, and it is plant diversity and crop diversification, diversification of income sources, agroecology, all these hallmarks of diversity actually are what our agricultural endeavors will be characterized, whether we are ready for it or not, because that's resilience equals diversity. That's just how it is. You can rattle on that tree as long as you like, and so I really am really grateful that we have in the plant world, for example, a diversity to work with that allows us to address challenges in the world, including climate change, and the ramifications that come with it. So we will be aware that droughts are everywhere and ever present, and they will actually in the upcoming future be one of our key challenges in New Zealand, and also in the wider perspective of the globe. So this model just shows the extension of this wedge into summer droughts from 25 days of intense heat to 50 by the middle of the century, and nearly by the end of the century, 75 days, which is nearly all of summer, just hot day after hot day after hot day. So that's just how it is. It's something we know and we can plan for. We are in uncharted territories. CO2 is one of those markers, and I would call it a marker of stress. The measurement is equivalent with those hotter days, with drier days, and so we can use that as a proxy for predicting what will happen. And then this trace here, the CO2 emissions are superimposed on population growth, and so and notice the blue and the red curve here, and so I've color coded the rest of the talk into this blue and red, and the blue, that's basically production. When you look at it from a plant science perspective, we need to make more food for all these people, and that's one challenge. The red line is the stress line, so that's our future as well. In addition to having more people, this food will have to be made under more and more stressful conditions. So we need protection in addition to production. Protection, stress protection is our challenge as plant scientists, and production, of course. Just as a nice little add-on, to that. So now we're getting into the plant side of the story. So plants basically have two big metabolic biochemical areas, how they work. The first one is called primary metabolism, and that is the production of the building blocks of life, like sugars, amino acids, lipids, and so on. And this all starts with photosynthesis, and that's why it's got that blue circle around it. That is the starting point of production. So that's nice. We know plants make all this food or represent all that food. The other metabolic reaction that we have is secondary metabolism. And so, okay, here it is again. So you've seen now that the primary metabolic area has been shaded, and we are now in that secondary space. And the secondary metabolic reactions, they are there to protect the plant. So in simple terms, we have a production part of plant function and the protection part of plant function. And so this protection part of plant function is characterized by a number of different compounds. They can be antioxidants, they can be sunscreens, they can be feeding deterrents, all sorts of functions that protect plants to make this beautiful primary metabolically driven growth. And so there is circled here in red, so that's now our protection side. One compound group as an example, the phenolics. So they are pigments, and they can absorb ultraviolet as sunscreens, but they can also act as antioxidants. We also taste them very happily in a red wine. And so they have all sorts of functions, and I will take them as a contrast to photosynthesis as a driver for growth. Here's our driver for protection as one example. And there is a trade-off. So Phil Grine is a very famous plant ecologist, and he developed a system that I was introduced to by my supervisor when I was doing my PhD, that you can either have production, which is of course what we need to grow more plants for the burgeoning world population, or you have stress tolerance. And so he developed this CSR system. C is competitors, S is stress tolerators, R are the rudderwolves, that's the plants that work well in disturbed areas. And so there's basically, you can be a stress tolerator, but then you're at the bottom of this curve. If you want to be a productive, very competitive plant, that's fine, but then you won't be in the stress protection corner, in the stress tolerance corner. So it's basically either growth or protection. And we have just seen in that graph before that both of the curves are going up. So if we go with Phil's system, then we're stuffed, because we can either produce more food, but that won't work with all the extra stress that will come, or we produce more stress-tolerant plants, but then they will be small and not going to nourish all these people. So that's the conundrum where we are trying to learn, by knowing how plants work, how we can move stress tolerators into a more productive space. And so another way to show this, blue and red again. Blue is production, red is stress tolerance. And so normally that green line here is what plants do following the CSR plant strategy theory, that the more stress there is, the red line, the less production you have. And so let's follow that one up a bit more. So in simple terms, it's a bit like a seesaw where the more stress, the less production. What we are aiming to do with the knowledge of how plants work is to move the triangle, the CSR triangle, from where it is at the moment, where it forms this pivot point for the seesaw, and move the whole triangle upwards in terms of productivity, so that our stress tolerators are now more productive, which then means that instead of having a pivot point for a seesaw, if you like, we can more equate it to a okay, I think I'll just go here now. The pointer is around it.
You want me to forward your thoughts?
No, that's fine.
Sure.
Yeah. So we've gone from a seesaw to a more what's like a springboard. If you look at the pivot point here is now not in the middle anymore. We're moving it upwards in terms of productivity. So it's more like a diving board, a springboard, so that green curve that goes down, thanks to the law of life called strategy theory, is now a straight line, and that's why it's called sustainability. We try and sustain growth despite stress going into the more extreme. That's our first mission, should we choose to accept it. So, do you think that's possible? No.
No.
Because there's a law of life called strategy theory. Well, let's see what has happened so far. And this is an ongoing story. So the toolkit that we can use for this is plant breeding. And as we've already discovered, diversity, genetic resources, are an invaluable tool to try new combinations, try new solutions. So I'll give you now examples from my favorite plant, white clover, and grasses in general. So we did a study where we looked in an experiment, and we grew plants from all the decades that had been collected by many scientists and stored in a seed bank in Palmerston North, and checked out whether there's perhaps a trend about time. Did we succeed as breeders, or the guys before me and the girls, over time? And yes, we've done well with productivity traits, but what is shown here, so here's red, here's blue, is that in terms of drought tolerance as a stress marker, the first half of the effort was actually quite good. And that was when we looked, or when scientists looked at wild material, plant material, as a starting point for a breeding program. So these so-called ecotypes, relatives of plants that come from the wild and then are used in plant improvement programs, resulted in increased drought tolerance over the decades until about the mid-60s. After that, you can see the curve go down again. And the reason for that is that in those later decades, the focus was in general, not in every study, but much more towards productivity. And so here's Phil Grime again. We got nice, lush, beautiful cultivars, but they are less drought tolerant. So that's what has happened so far. And so Cookie so far is right. Then we did a study where we looked at ryegrass, because you can't always do clovers, that's boring. And we found that actually there are ecotypes, so wild representatives of that species that are much more productive under drought. And who would have thought a Norwegian ryegrass is like 5 times or more better in growth under drought compared to a cultivar like Impact. So that tells us there's something in this ecotype story that we should explore. And in white clover, it's very similar. So this is a study of 120 different white clover lines. And in this example, we had nitrogen applied, or just relying on the nitrogen fixation of the ability of that legume. And what we saw, the more nitrogen there is, so that's nice and lush, productivity-orientated sort of growth, we get lots of shoot mass, root mass, whatever, compared to the other side, where when there's less nitrogen, then then that's the opposite of growth, basically. What the plants had showed us there was, oh yeah, we are really good in putting our effort more into the roots to get the sparse resources that are available and less into shoot. And so the root to shoot ratio is then the main characteristic here, which of course from a stress perspective is great, but from a productivity perspective is not great. So that's still the law of life, Phil Grimes. So then we looked at, okay, if there is ecotypes that could perhaps push those boundaries a bit, what could be done? And in Pami, they have the Margot Ford germplasm seed bank, where Margot Ford, a lady, a researcher from New Zealand, actually went to places like China. So this is in the Tian Shan Mountains, where white clover does grow at 2,000 meters elevation with a growing window of 2.5 months, and droughts and frozen soils. So they must be pretty resilient. And then hybridization occurred. So in breeding, one of the tools is to bring different birds together. We can do that quite easily in the plant world. So we then took the Tian Shan, which is the Chinese parent, together with a productive parent, so blue here, and we put them together, and then you get an F1 generation, which is like in humans, a mix of mom and dad, F1, and then you can actually, in the plant world, can't do that with humans, take then that offspring and breed it back into one of the parents. And what you do with that is you are then trying to introduce the new traits that you've created in your hybrid back into the desirable parent, the productive parent. So you have a chance to breed stress resistance into a productive plant. And so that's then called a backcross 1, BC1, and then you do that again, get a backcross 2. So that's enough theory. Let's look at some of the examples there. So this is from those Chinese mountains, northwest China. This is called Putu, which is a New Zealand cultivar, very desirable in terms of productivity. You can see that here. When you bring the two together, so this is now our F1, our child of the two parents, you get heterosis, which is that the offspring is bigger than the two parents. And so there was it doesn't always happen like that, but in this case it did. And so we then used this plant to test it in the field and see what it does under drought. And the first thing we saw in this F1, in this offspring, compared to the parents, was an increase in the root-to-shoot ratio. So that's good. But remember, we have a very productive plant here. It's bigger than the parents. And it has now a better root-to-shoot ratio, which is what we just saw in that other slide, a predictor for stress resistance. So that's great. We have started to make inroads into productivity and stress resistance. We then looked at those phenolics. Remember those secondary metabolites that are also stress predictors and stress protectors. And we saw that in Tian Shan, in the Chinese line, they were particularly high compared to anything else we looked at. So we then did a study where we drought stressed the plants, and we saw that the ones that were stress resistant against drought. So here's our axis, here's our starting point. Koputu is the parent. We have moved a number of these lines now in a territory where there was no reduction in growth under drought, and that was related to those phenolics, particularly increasing under drought. So that's an example of working within a species, and then you can have already introduced stress resistance. Now I'm going to talk about taking two species together, which is another way you can do this in plant breeding. So this is white clover. This is Trifolium uniflorum, which is a small, flimsy little thing. No farmer would ever be interested in this, but it's an ecotype. It comes from the wild, and it has stress-resistant properties because it survives in environments like this. And so when you then do another hybrid, this time it's a backcross 1, you get profuse amounts of roots, which is always a great predictor for drought resistance, and still the same growth habit as white clover does. And so my student Shirley then did studies in the field where we found a 2/3 increase of these phenolics, of these protective compounds in our hybrid. And we also already had seen that the roots were good. Let's look at what that meant in terms of data. So here's the cultivar. Here is the uniflorum ecotype. You can see already so much more roots. And then the hybrid is much more like the stress-resistant starting point compared to the parent that we wanted to improve upon. So much better productivity, and the same in COPU2. So we're creating, by using these hybrids, much more stress-resilient plants. Leaf damage was the same. So we had more leaf damage in the parent COPU2 under drought. So here's the drought plants, whereas in the hybrid, the leaf damage did not increase. And we saw the same with the roots, as you already saw in the pictures. So we had a maintenance of root dry mass. So not just more roots, but also those higher root masses were maintained during the drought, whereas as expected in the parent, the red line, it went down. So green means we're now improving on the blue and the red patterns of field rind. Green means sustained production. And the same happened with photosynthesis in these plants under drought. We maintained photosynthesis compared to the parent, which is the red line going down. So we addressed in this the challenge of maintaining under increasing stress, drought in that case, productivity, whether it's photosynthesis, root production, and so on. So it is possible. Just to show that it's not just the pasture plant thing, this is in soybean, where we also showed that compared to ecotypes, which are the black bars, we were able to show that you can have productivity. So this is high productivity in soybean, and that's paired with, compared to the starting point, less water use and more water use efficiency. So that's in the space of sustainable plant production where we are at. The next steps will be, if we know that photosynthesis is the start and these secondary metabolites are the end point, that we focus now on these pathways that link these two worlds of protection and production, so that we get a combination of those two worlds. And we also will use new technologies that are available that look at the whole plant in itself, all the genes, all the metabolites, the proteins involved, and the phenotype, the overall picture of the plant, to arrive at a new dimension of productivity that is not just productive but also stress resistant. The last 5 minutes, I will now talk about sustainable innovation in the Pacific. So what we've seen so far was sustainable innovation in a very nice, lush, beautiful, green, clean country. Let's see whether that can also be implemented in our neighboring region. So I'm working there with Australian research groups funded by the Australian Centre for International Agricultural Research, mainly in Melbourne, Lincoln here, the University of South Pacific and the Pacific Community, which is the major donor for research in the region. And we've written a report, and we are now in a 4-year project that is following up on that report, where we look at sustainable intensification. So it's the same thing, blue and red, brought together, sustainable, but more production. Can we do this in the Pacific? What do you think? So the key here is a concept called CASI, which is Conservation Agriculture Sustainable Intensification. And it is based on many principles of regenerative agriculture that is embraced to the core by that region. I was at the largest agricultural conference a few years ago, and the FAO, everyone came up with this final paper that said that this sustainable agricultural intensification based on regenerative agriculture is the only way forward, how they can get out of their disturbed soils and over polluted waterways. So this space of sustainable agricultural intensification has grown in terms of publications over the last few years. It's blossoming. And so what we did as a start was to go out to the farmers in Tonga and in Samoa as a large research team that included social scientists, biophysical scientists, and we created a survey that identified the needs of the farmers and the industry stakeholders for a start. And so they told us where the problems are. We then characterized these systems. We looked at the risk profiles and then developed options in the sustainable agriculture space. That's what this report was all about. We used mixed methods. So that meant we looked at qualitative and quantitative data, SWOT analyses. We interviewed 74 different farmers and several groups. Here are some examples of the locations. So that was in Upolu, which is the main island on the right, and in Savai'i, where Bruce Mackenzie's family lives. And yeah, we went to all these people, listened to them, and had our surveys to report back from here, the Nuku'alofa and Tongatapu locations. The challenges that were identified were a mixture of biophysical and social. And when we looked at particularly the biophysical ones that are listed here, a lot of it around plant, soil questions, but also technical knowledge, access to resources, and also access meant often women had even less access than men to resources, including sometimes field sites. And now the last few slides, I'll show you some of the interventions that we developed as the models that we are now testing in our 4-year project. So at the very beginning, yet again, diversity. Let's try new genetic material. Let's go and do that for plants and animals. Focus on resistance. As unbelievable as it is, that's still in the baby shoes, that sort of approach. And often is to do with resourcing. Crop diversity doesn't always just mean new stress resistance, but just different species. And so there's now projects underway that use that diversity yet again. Soils, a number of different approaches, nutrient cycling, a less disturbance-prone way of working with these very delicate soils. It's all possible and still grow more plants and more yield. Management. So what different types of management do I use? Mulches. What is the process here? You won't believe how little mulching is done over there. And yet we know that it has its benefits. Algae cropping is the same. So agroforestry for that matter. And it sounds like, oh yeah, this is airy stuff, but it's not. That's the only thing that works there. We can come with our conventional ways of doing agriculture, but that's already been done and it failed. It resulted in a downward spiral. Integrated livestock projects are in our work, and then IPM, improved water management, and then the more sort of social side. Lots of training is needed on all sorts of fronts, and that includes also agribusinesses and just using the right equipment in a hygienic way. And working also together with the local communities to provide a more equitable access to all these systems. So the next steps are now what we're doing at the moment as we speak. I'm going to travel there in a couple of weeks, is to go and validate what we have recommended. So we have what's called mother sites at the University of South Pacific in Samoa and in Tonga. And we have baby sites, which is where the on-farm testing occurs with farmers who are keen to contribute to that study. We will utilize all these different relationships that we formed over the years. Samoa's farmers organization, MODI, is the Agricultural Development Trust in Tonga, and the Scientific Research Organization of Samoa. This is the campus in Samoa. This is the one in Tonga. We will have field sites. You can see some here that test all these ideas, and that will include a variety of replicated experimentation that's quite fun to develop, and then to maintain with the people who are on the ground there. All of them are trained scientists, plus the baby sites. Here's one example. We have multiple different systems, monocultures, but also diverse cropping, and we'll align that with education and with the development of a resilience center in the Pacific in Tonga to pull this all together. So the last slide shows you some of the key players. Soane, who can remember Soane? He received the Lincoln Alumni International Medal at the last graduation. That's when he showed this. So he's the CEO of the MODI Tonga Trust, which is the Agricultural Development Trust. Prof. Beth Holland, she's a Nobel Prize winner in the international climate change space. She's also at USP. Tim Reeves, inspirational leader at the University of Melbourne in this space of regenerative agriculture. And then my direct collaborators here, who we have lots of interactions with, both this was a photo taken in New Zealand, and this is also when they remind me that the talk is finished. So thanks very much.
Thank you very much. We have time for questions from the floor.
Yes.
Rainer, you have done experiments here in New Zealand, nice and lush, and probably relatively easy. How did that translate into doing the experiments in the Pacific with the islanders and making sure that you got acceptance of your ideas?
Whoa, that's a very good question. So for those who haven't heard it, how do you even get buy-in for doing these fancy studies in an environment that is so different to ours? And it's a combination of efforts, and some of them are very much in the social domain. So, for example, I had never had kava before, but I had probably a lifetime supply of it before we even started to do any research. The culture is that you sit together and that you share this bowl of kava together, and that may be 3, 4, 5 hours. And there's not much spoken, and you just enjoy that. And you think you do that once, you do that sometimes 4 or 5 times a week. I didn't plan to do that. This was not my strategy, but I know for sure when I think back, that's the people I sat around the table are the people you've just seen on that picture, and we're still working together. So what I've learned, I did a sabbatical over there, and I had all the best intentions, 100%, but I was still behaving initially as a palangi, or pakia, as they would say here, just because, oh, let's do this. And they're like, I think he needs to drink more kava. So now it's, the people, initially there was a bit of a divide, and I noticed that, and I didn't know why and couldn't make sense, but now we have the most cordial relationships and these research projects. So I just needed to unwind a little bit of my expectations. And at the same time, we are all doing replicated field trials, and they are very good with those efforts. It's just the trust, the respect, that's the most important thing. Idea.
Do you think which is the major constraint, disease or drought?
So what is the major constraint, disease or drought? Okay, so I think drought will become more and more constraining. Disease is already a big factor. So the combination of those is mind-blowing. So at the moment, I probably would say disease, but in the future, probably more and more drought, as well as disease.
So you have a good support in terms of plant pathology system?
Okay, let's talk. So the lovely thing is the campus in Samoa has plant pathologist one. The one plant pathologist who also has to cover many other areas. They have one agronomist, one physiologist. So we think we are one deep in our discipline. These guys are really, and that's of course a resourcing thing. So the more we can work together, and then it includes students as well. There is a galore of projects available, and I'm 100% sure this attracts new funding. The MFED in New Zealand, the IFED in Aussie, they are all keen to support your research in this space where we're actually making a difference on the ground. Not just a quick fix and then we're out again. This is all about sustainability. So let's talk. And same for anyone else. Please come and see me after this. This is our backyard. This is where I think we can get so much satisfaction also in helping a region that really needs help. And at the same time, you learn so much, not just scientifically, but also personally.
Apparently I'm supposed to say it's pretty intelligent. So with your research in terms of the stress-resistant species and the lush ones, are you also going into looking at the how the stress-resistant plants are expressing and what pathways that is working through and what nutrients they're using more of, so then we know what we can put in to perhaps strengthen up the less stress-resistant plants in terms of compounds and...
Yeah. So the question is, when you know about stress-resistant plants and productive ones, can you make sure that you bring in all the traits that make a stress-resistant plant into a productive one? Is that
kind of? It's more, are you following the nutrient pathway that the stress-resistant plant is going down? So then perhaps if you were to add additional nutrients on plants that are not necessarily so stress resistant, see whether you can actually boost the stress response in the outside,
so it's
rather than replacing full pastures and that side of things.
Yeah, so it's optimizing nutrient pathways. Is that right?
Yeah.
So we haven't looked at that in particular. We're more at the moment at this relatively early stage, more looking at the plant composition, but it's a fair suggestion to just optimize nutrient uptake. For us, it's often a function of getting more root material into a productive plant that is used to not need that root material because it's getting all that nitrogen. So if we get an increased nutrient use efficiency by, for example, increasing the root to shoot ratio and still have a good shoot productivity, then that would be one way of getting more nutrients into the plant. But there will also be biochemical pathways that could be looked at for sure.
on a very practical layman's perspective, in our backyard and our veggie patch, how can we, I mean, these are fantastic principles. And obviously with the increasing weather events that are occurring in our neighbourhood and more broadly,
Yes.
How do we implement this into our backyard?
Right. So you mean as just a homeowner with a garden in the background? Yeah. Okay. So I guess it's just a matter of scale. So what we know from the work we've done is that you can increase the resilience of crops, sometimes by improving the crop itself, but sometimes you need to change. So for example, Cyclone Gabriel, there will be some crops we can't grow in certain regions anymore. The cyclone has just shown us that, and we can now pretend that there was a 1 in 50 years event, but the next 1 in 50 years event will probably happen. I don't want to doom and gloom things, but so then we might as well not grow those crops there anymore, or we grow them differently. That's another way of doing it. And so that would be applied the same in a garden sense, that you may decide I choose different crops or I protect them in a variety of ways so that they're not as exposed to the elements. There's also increased nitrogen use efficiency that you can introduce by having legumes that then you can co-crop with your non-legume crop, and so you will get some benefits from that as well. Yes, Paco.
Any thoughts in combining plant breeding and plant priming?
Plant priming, yes. Tell us more.
Oh, well, just training the plant.
Training-ish. Right.
Yeah.
So you stress it a little bit and then you
yeah, let's say the parental generation, so that information is passed through the next generation of plants.
Yeah, that's definitely part of that whole process of plant improvement. Yep. And it's probably not done enough. We often do more, we rely on our genetic improvement rather than using treatments to improve on the genes. Mm-hmm.
I'm sure that Rainer could talk to you all evening, and then perhaps we can do it outside with a beverage.
Yes.
But first, we'd like to thank you very much for this excellent series. Join me. So please join us outside. There's a little bit of food, some kai, and beverages. Thank you.